Good morning. I will be talking about development and succession of oral microbiome. First of all, we will look at microbial acquisition. It depends on mode of delivery, such as ingested milk or water, conventional delivery, conventional childbirth. In that cases, vaginal microbes from the mother are found in babies. Example: Lactobacillus, Prevotella, Aptobium, etc. In case of cesarean delivery, microbes which are present mainly on the skin that are found on the oral microbiome of the child. These are some of the Streptococcus species. Lactobacilli plantarum incorporated during breastfeeding that inhibits the pathogenic microbial growth. There is something called as vertical transmission and horizontal transmission. Vertical transmission is microbial transfer in successive generations such as Streptococcus salivarius and Streptococcus mutans, which passes on to the next generation is vertical transmission. Horizontal transmission is microbial transfer within same generation. For example, Porphyromonas gingivalis, Aggregatibacter acidomypotens. Okay. Pioneer community and climax community. This says the oral microbial growth, which can be seen in the oral cavity of individuals. So pioneer microbial community is the first microorganisms that grow and colonize until the development of environmental resistance. Physical factors such as desquamatization, chewing forces, salivary flow, nutrient availability can affect the pioneer community. Chemical factors such as redox potential and pH of saliva, antibacterial properties of saliva, etc. can also affect the pioneer community. Climax community is the dynamic colonization of microorganisms after the environmental resistance has gained the equilibrium. So this does not change and mostly remains as such. There is then we'll talk about allogenic and autogenic succession. Allogenic succession is community development influenced by factors of non-microbial origin. These factors are such as tooth eruption, varying denture, and antimicrobial agents used. Example for this is like marked increase in Streptococcus frequency with appearance of non-shedding hard surfaces, that is eruption of teeth, or insertion of dentures, removable orthodontic appliances, or use of acrylic obturators in cleft palate and cleft cases. Whereas autogenic succession, it's the community which development is influenced by microbial factors. Factors such as O2 consumption, creating anaerobiosis, food chains among the microbial community, or receptor exposure for attachment. Example for this is increased number and diversity of obligate anaerobes present in presence of teeth. Another example is food chain metabolic end products of one organism becomes primary nutrient for other organism. Now we'll talk about the microbial growth. Starting from the birth, mucosal surfaces are colonized by a species of Streptococcus family within a few days after birth. So protection from host immunity by IgA1 protease activity, it is shown by the microbial Streptococcus that prevents the immunity to kill the microbes. So Streptococcus species such as S. oralis, S. mitis, bovar 1 and S. salivarius are found by the first month of the birth. During teeth eruption, more species of Streptococcus are seen along with actinomyces. Gram-negative obligatory anaerobic bacteria, example Prevotella melaninogenesia, Fusobacterium nucleatum, Prevonella spp colonization in edentulous infants is seen about in 3 to 4 months. From age 6 months to 3 years, eruption has a significant ecological impact on the oral microbiota. Streptococcus mutans and Sanguineus A. Nestlandi are seen once eruption begins. Gram-negative obligatory anaerobic bacteria are isolated from gingival margin of newly erupted teeth. Members of the genera Neisseria, Veillonella, Actinomyces, Streptococcus, Lactobacillus and Rothia are commonly isolated in this stage. Porphyromonas gingivalis, Tannerella, Forsythia and Actinomycetomenon were detected in 18 months old infants. So, and periodontal pathogens such as P. gingivalis, Forsythia, Triptonema denticola are present in biofilms which increase over time. Then after 3 years, spirochetes and black pigmented anaerobes are found in 20% of the oral cavity by the age of 5 years. In adolescence, the number increases to 90% individuals due to hormonal changes in gingival cervix. Increase in Prevotella intermedia in plaque during 2/3 of the pregnancy is due to the elevated estradiol and progesterone. Increase in the number of black pigmented anaerobes has also been observed in women taking oral contraceptives. Attains microbial homeostasis due to interbacteria and host bacterial interactions during puberty. At age at a very old age like 60 and 60 above, so age related changes occurs as a result of direct changes in aging, which are like salivary flow is reduced or malnutrition and nutritional deficiency. So direct effects are cell-mediated immunity, salivary flow, and antibodies, hormonal changes, nutritional deficiencies, psychological alterations, physiological alterations. Indirect effects are like wearing denture, medications, cancer therapy, dietary changes, etc. So in denture wearing people, the albicans colonization is prominent, along with Tannerella, Forsythia, Porphyromonas gingivalis and other species, at the mainly isolated from the dorsum of the tongue. So higher frequencies of lactobacilli and staphylococci and yeast are present in the saliva of healthy subjects of age 60 plus. Decrease in IgM and IgG antibodies causes increase in susceptibility to disease and also the microbial growth. Carbohydrate rich diet increase acid fermenting and carious bacterial growth. Smoking is associated with the increased prevalence of Parviomonas, Fusobacterium, Porphyromonas, Treponema, Campylobacter, and reduction in Veillonella, Neisseria, and Streptococcus species. So the common distribution is as seen in this figure. Uh, in cheeks, lip, and palate, there is despermatization seen. So the saliva is the major influencing role, and microbiota has low diversity in these regions. So mainly they are facultative anaerobes which are isolated from cheek, lips, and palate. Some periodontal pathogens persist by invading buccal cells. In teeth, non-shedding surfaces, stagnant sites, food impaction is possible. So there diverse microbiota is seen, and it is site variation, site dependent. Many obligate anaerobes are also seen, influenced by saliva and GCF. Species seen such as Streptococcus, Actinomyces, Veillonella, Fusobacterium, Prevotella, Treponema, and some unculturable organisms. In the tongue region, it is a highly papillated surface, so some anaerobic sites presence, and there are anaerobic bacteria seen such as anaerobic bacteria are also seen, and the tongue region has a diverse microbiota. So species such as Streptococcus, Actinomyces, Rothia, Neisseria, and some gram-negative anaerobes are seen. So what are the benefits of oral microbiota? The natural resistant oral microbiota provides essential benefits to the host, including colonization resistance, that is the pathogenic bacteria cannot colonize due to the presence of the healthy oral microbiota. Crosstalk with the host, modulates immune responses, upregulates beneficial host pathways, enhances mucin production and nitric acid. It also helps in nitrate reduction to nitrite, and nitric oxide. So the commonly used method for determining oral microbiota composition, it is either culture methods are used or molecular methods are used, which is culture independent. So basic steps are like sample acquisition, then the transport of sample and dispersion of sample. Then the sample is cultivated, and enumeration and identification is done using microscope or in situ culture or molecular analysis. So in culture methods, these require the growth of microorganisms on non-selective and selective agar plates under appropriate conditions. Only 50 to 70% of the culture of microbiota can be cultured, whereas in molecular methods, metagenomic approaches enable the detection of both culturable and non-culturable microorganisms. The function and activity of oral microbiota can be determined by metatranscriptomics, that is RNA profile, metaproteomics, protein profile, or metabolomics, that is metabolic products that are produced. So these way we can identify the microorganisms that are found in the oral cavity.